Very good morning, Konanda. I am Dr. Nagalma from Department of Pharmaceutical Analysis at SNS College of Pharmacy and Health Sciences, Coimbatore. Today we are going to do this DT-based learning about different types of pharmaceutical analysis. In the pharma industry, we are doing different types of drugs manufacturing like solid dosage forms and liquid dosage forms we are doing. After finishing that manufacturing or a preparation, we need to check the quality of the product. So we need to give this confidence to the patient when they are consuming this right drug at right dose, they will get health benefits. So that's the reason we need to focus always about safety, quality and efficacy. So if you want to get the safety, quality, efficacy from the pharmaceutical formulations, we need to check or we need to perform the suitable quality control test for the formulation. These would be quality control tests we are doing during the selection of raw material and after that bulk processing, and finally the final product also quality we need to estimate. Then only we can give surety for the formulation and at the same time the product is safety for your patient. So in this DT learning today, the design thinking based learning, today we are going to learn about what are the different types of pharmaceutical analytical technique we have. We are going to do the discussion and we are going to learn. In this overall, this quality control technique, we can classify three categories. One is spectroscopy method, like ultraviolet spectroscopy, UV-Visible Spectroscopy like, and another one is chromatography technique. In this HPLC Thin Layer Chromatography, paper chromatography, high performance liquid chromatography, and HPLC, high performance Thin Layer Chromatography, and other UPLC technique also we are using. And the third category, we are performing in this electrochemical method of analysis like potentiometry, conductometry, polarography, amperometry technique. Even my students going to share their views and the different types of quality control technique in the pharmaceutical analysis.
I talk about different types of titration. There are main types of titration: acid-base titration, redox titration, complementic titration, precipitation titration. This is also involved in different types of titration.
Sure.
I'm talking about analytical techniques in pharmaceutical analysis. Different types of techniques in pharmaceutical analysis. I'm speaking about electrochemical analytical method. Electrochemical analytical method types are polarography, potentiometry, conductometry, amperometry, and etc. Potentiometry, which involves electric current voltage and refractive voltage. In polarography, which refers current flows in the volume, and in conductometry, the electric conductivity in the current, which refers in different types of electrochemical analytical method, and also conductometry and voltage and amperometry, which involves with current, which involves volumes of amperes in the different types of electric analytical methods.
Then now we have to talk about spectroscopy methods like UV, IR, NMR and mass spectroscopy are essential in pharmaceutical analysis for identifying compounds, determining structure, quantifying components, ensuring quality control. They work by analyzing the interaction of light or other electromagnetic radiations with matter to provide detailed information about molecular composition, structure, properties. These techniques are used throughout the drug development process, from raw material inspections to final product testing.
Hi, I am Praveen. I will tell about Chromatography in this method. Chromatography is vital analytic test in the pharmaceutical industry used for the separation, identifications and qualifications of compounds in the drug samples to ensure the quantity, safety and efficiency. The keywords are including the purity test, identification in impurities or contaminated drug development, isolated active ingredients, quality controls, ensure the batch and stability. For example, thin thin layer chromatography, paper chromatography, and in high performance liquid chromatography and high performance thin layer chromatography, and in gas chromatography.
So my students, listed out few spectroscopic method and chromatographic technique also. Even I would like to add few points here. This thermal analysis method also playing vital role in the quality control department. So for the specific API, for the qualitative analysis, I mean kind of identification purpose, we are using this thermal analysis. The TSC method is playing vital role, like differential scanning calorimeter and thermogravimetric analysis. Based on this melting point of the API, we can identify or we can estimate the quality of the product or the purity of the particular API. The API, I mean the active pharmaceutical ingredient, also playing vital role in the drug manufacturing. So when we are selecting the right and safety and high purity compounds of API we are selecting, we will get the final product also with high quality. If you are compromising in this API quality, we cannot expect better quality in the final product. So this API, active pharmaceutical ingredient identification, the thermogravimetric analysis and differential scanning calorimeter also playing vital role. I hope this video is useful for the B Pharm learners. Thank you. Have a nice day.